Good afternoon. Hope you won't fall asleep during this talk. I know you're tired and I'm in the end, but again I will also use the ability to thank for the invitation and for the nice lunch we had here today, so that was very good. So I'm going to talk today about East Greenland polar bears and see if they can provide us with some information about the contaminants that we are all being exposed to in the entire world. Let's see if I will succeed. Yes, just to tell you that like all good science, it's a function of collaboration with a lot of colleagues, and two of the people present here has provided funding, not funding, but samples from from Svalbard and Canada that I'm going to touch upon. So thanks to them as well. During our discussion yesterday, we talked about the multiple stresses, the threats towards the polar bears, and we've heard of quite a few of them. We've talked about hunting, tourism, and oil exploration and climate change, but I'm primarily going to touch upon the contaminant loads of the polar bears and how that affects the health of the polar bears. So these are the three items that I will touch upon, mainly. So here we see one of the ear colors that June talked about before, that has been used on the males, that provide a little shorter information than the colors used for the females. And just to give you an idea of the distribution of the East Greenland polar bears, they're distributed along the ice coast, not too far ashore, and that's of course the limit you see here is mainly the ice edge that you have seen on several other presentations. Here we actually see that there's a partial overlap between the Svalbard polar bears and the East Greenland polar bears, but management-wise they are separated. We also on the east coast of Greenland we see a seasonal separation. So you see that during winter you have some of the yellow areas, and if we go a bit more into details on that, it actually reflects that the females are going in and are denning in various regions in northeast Greenland and in central East Greenland, and there might be a region here as well that we didn't get with those few animals tagged. Well, I don't gonna dwell with this picture too long because we have had some excellent presentation in the past about climate over time, and we heard from Andy that 2/3 of the polar bears has been estimated by the US Geological Survey will be gone in another 40 years. So that's just to set the stage with the climate change. But we do, with contaminants, they're being globally regulated. And if a substance which are, I mean, we're talking about thousands and thousands of substances that we are all containing. And in order to regulate those things, we shall be able to prove that they show long-range transport, that they are chemical persistent, that we can indicate a threatening temporal trend, that they are showing biomagnification and has toxicological effects. And again, in the Arctic region, we have a very important reason for looking into the exposure of all the wildlife is because the Inuit population is feeding on marine mammals to a very large extent. Well, when you see here, this is the mercury emission from the AMAP assessment. You can see that mercury is mainly coming from East Asia, about 65%, is coming from China and other Asian regions, where you see no mercury coming out from Greenland, for instance. And if you take another substance like the PCBs, that was has been reduced around during the 70s, depending on various countries. Then again, you see they haven't really been used in the Arctic region. So now that you saw the distribution of the polar bear on the east coast here of Greenland, you'll say that man, he's crazy. I mean, there's no way these substances are going to end up in the polar bear. Well, the theory is that these substances, there are different pathways for different contaminants, but many of the persistent organic pollutants has a very high vapor pressure, which means that they are getting up into the air, and then they're precipitated as they get to the colder region. It's a process we call the grasshopper effect, and so these contaminants seem to move towards the northern regions. Another thing is that if we just take a simple substance here, like the PFOS, that's a perfluorinated compounds. It's some of the surfactants that are being used these days. They're not fat soluble like most of the other persistent organic pollutants, but still you see there's a substantial biomagnification taking place. I mean, we're talking about the order of 6 to 8 what's called not decades, but orders of magnitude, the increase going up the food chain. So that is quite substantial. So like we talked about yesterday, despite that loads are lower in the Arctic region, then still the loads are extremely high in some of the high trophic species of the Arctic. So when we're working with polar bear, I know this is a busy slide. You're not able to read all these different species, but it's you must just trust me that the green area here, that's the terrestrial environment, and once you get into the blue and you get up the food chain, then you see an increase. And again, we have a logarithmic scale here. So the red area is a different polar bear population, which is in the area. All these examples are effect levels that has been shown in other part of the world, either in the Baltic ring seals, where about 70% of the population was sterile, but there's also been studies on mink carried out and mice and other things. So the only animal that actually is exceeding the polar bear in concentration-wise is the killer whale, and these killer whales are from the Pacific. And right now we are getting in samples from East Greenland, and due to the geographical pattern, we expect to see even higher loads for the killer whales that has been predating on marine mammals from the east coast of Greenland. Well, here we see some samples from here. We have heard about the management zones, and that makes sense when we are looking at contaminants, that we use those information when we distinguish between samples taken from various areas. So this is mercury loads in hair from polar bears. We heard about hair sampling before. So that's one of the things that hair can be used for. And here we see a very distinct geographical pattern where we have the highest concentration in Lancaster Sound and northwest Greenland. And actually those patterns are pretty consistent if we look at mercury loads in livers or in kidneys of the polar bears. So we see the exact same pattern. But if we look at the Svalbard region, we see quite low mercury loads, and we on the east coast of Greenland also see quite low mercury loads. So those populations are in a good status relative to the mercury loads. But if we look at some of the persistent organic pollutants, we can see that the pathways are completely different. So if we look at the some PCBs here, then we see the highest loads on the east coast of Greenland and at Svalbard. And we have taken up the challenge because on the east coast of Greenland the polar bears are being hunted. So despite our Norwegian colleague has done a lot of good work on looking at samples from blood, we are looking underneath the skin of the polar bears and looking at histopathology of various tissues that I'm going to touch briefly upon later on. Well, what can we use the hair samples for? The first 20 years I was doing work, we only look at the tissue that was eaten by the Inuits. But we realized that if we were supposed to say anything about pre-industrial contaminant loads, we had to look at other matrices. So here we start looking actually at hair, and since hair is a non-invasive tissue or matrix, it's actually not so bad to use hair, and hair is also used for evaluating toxicology for humans, because not for obvious reasons people don't want to have a liver sample taken or other invasive things. But here we have taken sample from the National Museum of Copenhagen, where we managed to get us back before the 1900. And actually when we started out doing that, we realized that we really couldn't use the samples because they had been preserving the samples at the skin samples with sublimate, which is mercury chloride. So we came up with some ancient results about 20,000 times higher than the recent. So we actually managed to solve this by looking into the organic mercury of these samples, because hair is entirely organic mercury in polar bears. Here again we see the Svalbard loads of mercury are very low, and on the other hand the Lancaster Sound is extremely high, and we see a significant trend over time. Okay, this is a slide I've taken where we looked into other hard tissue, getting us back in time. So we were thinking that okay, maybe what we're seeing in the polar bear is an artifact, but can we actually show a similar picture for other things like birds of prey feathers, human teeth and ring seal teeth and other samples? So since the concentration were quite variable, we said, okay, present concentration is 100%, and if we're going back in time, what percentage of the ancient samples averages were it relative to present concentration? And that actually showed a very clear picture that the onset of the increase of the mercury increase took place about 1850, which is about where the industrialization started off in the Western world. And since that time, the increase has been approximately 20 times, which means that it's very likely that 94% of the mercury loads that is being carried by Arctic top predators are being created by some human activity. So for that reason, it's highly relevant to look into whether there are effects linked to those loads. We sticked our neck ahead in a publication from 2011, and since all the climate change people, they get the world's eyes upon their predictions by looking into the future, and we heard that all models are linked to have problems, but here we have tried to make projections 100 years ahead, and looking into the various wildlife effect concentrations, which are actually not too convincing. There needs to be more work done for relevant species, but we can see that we are not far from the various effect levels. But actually we have looked into to the brain, because you know that mercury is affecting the brain. So this is an early study by Basu et al. We did in 2009, where we looked at different biomarkers for brain activity, and here we did find, for instance, a significant negative relationship with the NMDA receptor levels and the mercury loads, both on the total mercury loads and the methyl mercury. So that means that the brain is probably working less efficient if you have a higher mercury load, and actually at those concentrations that we see in the Arctic, there seems to be an effect. We are now going into other parts of the brain, so those data I showed you before is from the brainstem, and actually the brainstem is the lowest part of the brain. So if you go into the pituitary gland, you have almost 5 times as high concentrations of mercury. So the likelihood of you have effects in other parts of the brain is quite likely, and we will probably have data on that the coming spring. So we're looking forward to that, and here you see the various parts of the brain that we're dissecting out. Well, the good news is actually that the polar bear, you would think, I mean, if you look at the liver levels or kidney levels, it's the highest mercury loads of any mammal in the Arctic. But actually, when you look into the brain and compare it with other species, then there's actually other species that are higher. On this, here I show you the threshold, the normal realized loads for neurological effects, and actually the polar bears are below those. But there's another group of marine mammals that are in that range, and that's actually the toothed whales. And what is the explanation for that? We think that when we're monitoring the mercury in hair, then the concentration is about 1/3 of the concentration in the liver, and maybe Andy knows more about that, but I've been weighing quite some furs or skins of polar bears, and a big male is likely to produce 6 kilo of skin per year, which means that they can clear 2 kilos of liver totally from mercury. So they're excreting a lot of mercury through their hair, and like you know, whales don't have too much hair, so they don't have that ability. So that's probably the reason why toothed whales have so high concentrations. This is effect levels that I showed you before from the polar bears. So now we are moving a bit over to the persistent organic pollutants, and I told you before that PCBs were regulated in the 70s, and like you see here, this is on juvenile bears. We have seen a decline, and that's the paper that we just heard about that came online a week ago. where we also see the same, exact same picture for DDT and actually for a lot of other substances. This is just, I'm not going to drag you through all these graphs, but it's just to show you that the juveniles are here. This is one of the PCB congeners. I showed you the sum of PCBs, but there are actually 209 different PCBs, so we're showing the most important of those in the present article. But we're also showing data for, this is from juveniles, but we also show for adult females and adult males. And what you can see is that if they're blue, the lines, then the trend is not significant, so that proves that juvenile animals are actually better to use for monitoring temporal trends, but that's partly also due to we do have a little more samples and a little more years than with the adult animals. If you look after year 2000, you could, if you're still awake, you might wonder that what's happening here, because it seems that the decline has sort of stopped after year 2000. So the last 10 to 12 years, there hasn't really happened too much with the PCB loads. And why is that? If we look at the biomagnification rate here, this is actually the concentration in ring seal versus the polar bear. Then that biomagnification has increased over the last 10 years. So that sort of proves that something is taking place there, which may not be linked directly to the human processes and the long-range transport of these contaminants. So something is taking place within the biological systems in these Arctic regions. Here I show you the stable isotope levels. That is an indication of the increase. So something is taking place there for sure. But we were lucky to collaborate with Melissa McKenney in Canada, who is presently working up samples on the East Greenland polar bears. And there she was actually able to look at the fatty acid composition going back to 83, almost 30 years ago. And what we can see here that the top graph here is the ring seal. So you can see in some years ring seal might constitute as much as 90% of the food, but it's actually varying quite a bit from year to year. And another species that is of quite large importance here on the east coast of Greenland is harp seals. And actually also hooded seals. So they seem to take over, or the polar bear are moving towards a higher proportion of these two species, which actually fits very well with the ice edge is moving closer to land, so the polar bear can actually access the breeding patches there on the west ice. Not all contaminants has the same good story like the PCBs and DDTs. Here I show you some data of the PFCs that we published in 2008, and here for all the 8 PFCs that we were reporting, we saw a dramatic increase. For the brominated flame retardants, we have seen increase, but that seemed to peak around 2004, so that's another good news that actually we've started to decline, and that's also part to human regulation of some of the brominated flame retardants. So does this actually reflect the production of these various contaminants? I would say yes, it does. If we look at the world production of PBD 47 here, you see that the peak here on the polar bears is actually coming approximately 1 year after the world production of PBDs had started to decline. So actually the polar bear is a very good sentinel species for monitoring these changes. And then, of course, it would have been good if I could have shown you a similar time trend for some of the other contaminants, but I must admit that I started taking samples. I've used half my life taking samples of polar bears, so I was 30 years older or younger at that time here. I went around together with the Inuits and on the dog sledge. I used a couple of years of my life on a dog sledge. So that's where we started to take the samples, but I mean, that's not good enough. I mean, I should have started earlier. So why didn't I start down there? And I must make an apologize for not doing that. But this is how I looked like in 1958. I'm sitting here, and of course I was working with polar bears here. We were also driving dog sledge. We were staying in the igloo during night, but unfortunately we didn't have enough polar bears in Sweden to take, and we didn't have current in that house, so we couldn't store it. So either I haven't been focused enough, but I think this gives you a good idea that if you want to get a picture and explaining things, you really have to stick with the problem for a very, very long time in order to observe the pattern. I know that there are people like glaciologists that are much smarter than we are, that can get millions of years back by just drilling a hole. But you should call me when you find a polar bear in your ice cores there. So this is another heavy slide that shows that the pattern was pretty consistent. So for all the conventional pops here, and all the 3 different age and sex groups, we see significantly declines for all the conventional pups, which is good news, I think. With the brominated frame retardants, the picture was exactly opposite. So we have significant increases over those 30 years, but some of them have luckily started to decline. So, of course, what does all these contaminants tell us? And this is a way of actually ranking the different contaminants and actually adding their effects. Because that, if you have such a mixture, you need to add the effect in one way. Some are more toxic than others, and it's of course you have to define what toxicity you're talking about. And there's a way of doing that by risk quotient measurements or calculations, but that's based actually on mice. So you would like to have those results from the same species that you're actually working with. But this is the effect relative to reproduction. But what is interesting here is that the risk quotient is actually 100 with those loads that we have been analyzing here. And what you can see is that the blue line here is the sum PCB. So when we're talking about effect on reproduction, the PCB is the very, very, very bad substance. Whereas if you're discussing whether could we use a DDT in Africa, then some might say, well, that may affect the polar bear badly. But as you see here, the DDT, at least relative to reproduction, doesn't have a really large effect. So that's the kind of way you can use these things. And here, you know, this is not just for 1 year. Again, we have tried to estimate the sum of all these contaminants over the 30-year period. And what you can see is that, you know, I showed you that some of the substances are increasing and some are declining. But the overall picture is that, you know, over those 30 years, the effect of reproduction is actually quite high. So, and if you then start to take the effects of climate change, where the animals are starting to use their fat resources in another way, that might add on to the stress of the animals. Okay, yeah. This is just an example of some of the work that we have done on reproduction, and I'm not going to take you through all the histopathological slides, but this is just an example of the length of the testes of the polar bears, and we have shown that those show a significant decline with the contaminant load, and similar results were actually documented for the ovaries, and that may reduce the quality of semen and eggs. But again, to get from here to effects on a population level, we still need to do a bit more work. But we have actually now collected live samples, and we are working with colleagues from the Pharmaceutical Institute to make dose response on all the cascades of sex hormones in the cells. So we are looking forward to those results. So this is one of the final slides where we have made a similar exercise on the effect on the immune system. And there again you see that that some PCBs is really the big bad boy here. It's really driven, that effect on the immune system is driven by the PCBs, some PCBs. But second largest is actually the PFOS. And some recent result has actually shown that there is an effect on the immune system on humans at the Faroe Islands. That came out this year by Grandjean et al. And if you look at the concentration lows at the Inuits in Greenland, I've just taken a small selection here, then at least some of the populations are even higher than the population on the Faroe Islands. And if you look at the polar bears here, the juveniles and the adults, then they are way above the contaminant loads for this group of perfluorinated sulfone amides here, which is PFOS and PFOA. So there's a reason to look into the effect of the immune system, and we're doing that. And the Norwegians has done that before and actually shown effects on the immune system. So future, we have a lot of things what we want to do. We have collaborated the last couple of years with colleagues. We've invited colleagues to East Greenland, working with these polar bears from Norway, Canada, and the US. And we hope to, if we can get funding, we want to look at other areas of the Arctic. Could be interesting to go to the Kara Sea. It would be interesting to go to Hudson Bay and look at climate-stressed polar bears. But we also have an ambition of looking at some of the pilot whales on the Faroe Islands and ideally also killer whales. And in Denmark, there's just been started the Arctic Center there, and that has also a lot of ambitions of trying to link the climate change, for instance, with these effects and of the chemical contaminants. And when we're doing quite a bit of analysis right now, we have a program called Oceans 12 that will come out with results on that, and we are doing a lot of work on those interesting time series. So yeah, one of the ambitions here is the combined effects. So to finish off, I want to acknowledge some of the funding agency, and I'm proud to say that we actually managed to get some support for Prince Albert too, who is also part of this institution here. So they paid for some of the work where we did all these time series, and that was done during the International Polar Years a couple of years ago. Thank you very much.